Welcome back, everyone. Our second speaker is Dr. Lance Osborne, who is a professor of entomology and associate center director at the University of Florida at Mid Florida Research and Education Center in Apopka. Dr. Osborne has dedicated his career to studying biology and management for invasive pests and ornamentals grown in protected culture, focusing on biological control strategies for thrips, sweet potato whitefly, mealybugs, and spider mites. He holds U.S. and international patents on biological control of whiteflies and other pests using a fungal pathogen. Dr. Osborne has served on two technical advisory committees, a National Q Biotype Whitefly Technical Advisory Committee and the National Chili Thrips Technical Advisory Committee. He has garnered national and regional recognition by industry organizations such as the Society of American Florists and the Florida Nursery Growers and Landscape Association. Lance, the floor is yours.
Thank you. Appreciate being invited. Let me see if I can get my talk going here. Everybody see that?
Yes.
Looking good, Lance.
Okay. I'm going to get started. I have probably more to say than I have time, so I'm going to go through this the best I can. I appreciate the opportunity. Just before we get started, I will show this at the very end, but the QR code takes you to a website on Thrips parvispinus, which is going to be the topic of my discussion today. So you don't really have to take notes. You can get most of this information off of our website, which is a combined effort of a lot of contributors, some very good entomologists that are working on this particular pest. No particular order here except alphabetical of the people that are involved. This has been supported through a research grant from USDA, ARS, FNRI, and then a little bit from Endowment Research Fund that got us started on tracking the spread of this pest. Okay, Thrips parvispinus, its origin is tropical in nature from Asia. We think probably in India and in that area. It's been detected in USA, Puerto Rico, Canada, Europe, India, and other countries in Asia. Parvispinus has been found in Florida, Georgia, Colorado, North and South Carolina, and it's probably been detected in a couple other states that we're waiting for USDA APHIS to do a final read on what's been sent to them and to corroborate what we think are other states. There are a lot of synonyms. When we first started talking about this pest, everybody had a different name for them, and because the parvispinus isn't that easy to say initially, we had a lot of people that wouldn't call them by parvispinus, and a lot of confusion of what we were really talking about. We've settled down. It appears that some people are beginning to call it pepper thrips, but we are still sticking with calling it parvispinus by its Latin name, so we know exactly what species we're talking about. Luckily, we haven't detected any cryptic species or any biotypes, at least to date. So it's generally a uniform group that we're talking about at this point in time. It's polyphagous, so it feeds on a lot of different things. It's There's a real problem with detecting host ranges with some of these insects, especially with thrips. They may be found on a plant, but that doesn't necessarily mean that they're feeding on it or that it's a reproductive host. A wind may blow them onto a plant, any number of ways that they may end up on a plant, and then if they happen to be in the reach of entomologists, they get collected and they get blamed for feeding on that particular host, which may not be the case. They can be very finicky. We have some colonies in greenhouses where we have papaya and they're not feeding on the papaya. Now, there's no flowers on the papaya, so that may be part of it, but it's not really a good host. It doesn't seem to take any interest in that, whereas it was reported to be a major pest in Hawaii on flowering papaya and feeding on the fruits as well. But it does get on enough ornamental and vegetable crops to cause us some significant worry just how big of a pest this thing might become. There are certain hosts that we find them consistently on in commercial nurseries and retail outlets, which we'll talk about as we go through this talk. It was first detected in a greenhouse in Apopka, which just down the road from here, near where I live. That was a greenhouse that was bringing in anthuriums from offshore, and they feel that they either got the thrips on the anthurium or some Hoyas that they were bringing in as well. We found it in the field on Cocoloba, eggplant, gardenia, Hoya, Asclepias, beans. Diplodenia has been hit particularly hard. Peppers in the field have been hit extremely hard. So the list is probably going to grow, and it may even shrink in some cases where we find that they really aren't feeding on some of these hosts. That's one thing that we're looking at, and a number of other people as well, to find out what is a good host, what is a reproductive host, and which plants we need to be most concerned with. The probability of establishment in Florida greenhouses is high. We already have good enough evidence that it is spreading through the state and establishing in more northern counties, which I'll talk about in a second. There's a couple distinctions. One is a detection, and a detection in nurseries or retail outlets doesn't necessarily mean that county that they find it in say a Home Depot or Lowe's, doesn't mean that county is infested. It's just a detection. And if it's in on a potted plant that's for sale, they don't list the county as being positive. Whereas if it is found in plants that have been planted in the landscape, in this case, this happens to be Oz, the person that was running for elected official, his house in Palm Beach. It was heavily infested. And Palm Beach became our center of interest, where we're starting to see a lot of plant material in landscapes that concerned us with the proximity of these infested areas to what was the main growing area for in not Central Florida, but down in Florida near Barrel Glade. This is a major area where peppers are grown. We warned the agents in summer that we had concerns that they're liable to spread. And within a few months and toward the end of November, we found the first spread of this particular pest on peppers in the field. And that particular grower misidentified it, didn't really know what was going on, and ended up losing his entire crop, about $1.3 million loss. At this time, DPI was saying the most northern range of this particular pest was right around here, Central Florida. This is the research center here. Disney's right down in this area here in Four Corners. And this is the farthest north that it had been found. But considering what we had found as part of a grant with the Florida FNGLA, we looked in Palm Beach County to see where this thing was and how it was spreading. And we looked at a number of retail outlets in this area and found that every one of the retail outlets that we went into, with the exception of one, was heavily infested with the not chili thrips, but parvispinus. So we know that they were spreading. We found them on Diplodenia. We found them on peppers and a few other host plants. So again, we know that that they're moving. Whoops, excuse me. I'm trying to get here. They're moving on Diplodenia, gardenia, and peppers. They were fairly consistent. And then we started picking up some new host plants, Plectranthus, Ruellia, and Vinca. 93, which basically all 13 out of 14 were infested. So we went, a group of us went up this is changing on its own. I don't know why it's doing that. We basically went up the I-75 over to I-95 and back down again, sampling for chili thrips in the, got thrips on my mind, I'm sorry, parvispinus in just about in every single one of the nurseries that we looked at along this and back down I-95 to Palm Beach. So we could basically find them at will in retail outlets, which caused us a significant amount of concern because you can see most of the state has them in retail outlets. That doesn't mean that those counties are infested yet, and that's one of the things that we're trying to do now is enlist master gardeners and anybody that's willing to cooperate with us to see if we can find them outdoors. And why is this important? Because if it isn't considered widely distributed and established, then it is considered a regulated pest. And a grower, if he's found to have these thrips in their greenhouse, they will put a stop order on their plants. They will not be able to move anything out of those greenhouses until the regulatory officials go in and don't find any thrips in there again. So it's a negative inspection is the only way you can start shipping plants again. We know that it's been found outdoors and established in landscapes in 7 different landscapes. Okay, so there's one up here near the research center, and then down along the east coast of Florida. Thrips management starts with scouting. And this is going to be important, especially for those of you in Georgia, because it was found in one retail facility. I think it was a grocery store on plant materials that could be planted outdoors, but we haven't found it in any other facilities, any retail outlets, or in the landscape yet. It doesn't mean that it will. We just don't know what the biology is of this pest enough to know if it can withstand over the winter in Georgia, North Florida, or any farther north. We know that it will survive in the winters because it is a major pest in Canada, but we don't know about outdoors. In order to identify these particular thrips, you need a very good hand lens. They're much more difficult than some of the normal thrips we deal with because it's the smallest thrips that we have in the state of Florida. We thought chili thrips was bad enough. Okay. When we started looking at chili thrips, the only way we could go find the chili thrips consistently would be to go up to the plant material we're wanting to sample and to use a beat board, or in this case, is a clipboard with white paper. You knock the foliage against it, and then you use your hand lens or some sort of magnification to see what's crawling around on the paper. The parvispinus is about half the size of chili thrip, so it's extremely small. You just see little tiny black things moving on the paper. They can be confused with columbella because they jump around a little bit and they're about that size. Okay, we've had a hard time getting some of our growers and landscapers to do this. One problem, as you can see here, my biologist is getting her hair down in the middle, so she's picking up thrips, and it's hard to really get them identified because they're moving around so much. So we do have some issues with this being that successful in finding initial populations of the thrips. The other issue is collecting them for identification. They're very skittish. Even in the greenhouse, when you just walk up to the plants, it's like they can sense you're there, or something sets them off, and they run on the bottom of the leaf. In this case, Joe Funderburk, one of the premier entomologists, with thrips researcher, he basically just carries a little tiny vial in his hand, and then he'll use it and dip it, and then put it onto the thrips so he can get them into the vial so they can be sent off for identification. We hit on another way of doing this that seems to work fairly well for us, at least collecting or getting some samples to send off for identification. We'll take a dinner plate, which we're using as our beat board, so to speak. We smack the plant against it, and the thrips, in some cases, would stick. But we found out very quickly that not all yellow sticky cards are made the same, or all sticky cards are the same. In this case, the thrips got up and walked off. We really didn't get any of the parvispinus to stick to the yellow sticky card. The blue sticky cards, on the other hand, are a little bit more, they're more tacky. These particular brand are sold so that not having sick them so bad that you have to go after handling them to go in and spend a while trying to get the stickum off you. So they're not nearly as tacky as some of the other commercial ones. Other yellow traps maybe work perfectly fine. But we tried to put a trap on this plates as another way to sample. So we'd hold the plate and then we'd slap the plant against it. Now you can see, even though they're not that tacky, they will catch the plant. But you can see that a lot of these little black things crawling around on the plate are parvispinus, and these are probably the little yellow ones are probably males. You can stick your finger on it and get your finger a little tacky, and then you can catch the thrips and stick them back on the card. And we could use these and bring them in to identify under microscope to make sure they're not one of a couple other potential thrip species that we can confuse with parvispinus. Okay. This is one of my colleagues, Dr. Ahmed, put together a field guide. This is on the bottom of a white piece of paper that you could use as your beat board, and you can compare to the size of the ones that are on this paper. This is the parvispinus, and this is one that's a chili thrips. Now they're all dark in color except the chili thrips, and so there are a few that we can confuse for being parvispinus, but in general, you can look at the banding on the antenna with the hand lens on the sticky card to determine whether it's one of another thrip species. This is parvispinus, very tiny, and it's hard to see the bands on it, but the bands are distinctive. And again, if you have any interest in trying to get more detail on how to separate these from others, please let us know. The best way would be to send them to extension agent or to you've got Eric Schuler there in Georgia now, who's an expert on this particular species and looking at biological control. These are the females on, I think it was probably eggplant. They're very tiny. They're very skittish. They don't like to be looked at or photographed. This is a male. Now that could be confused for a chili thrips. So again, it's very thin, very skinny, and very small relative to chili thrips. Okay. It's important to know a little bit about their development and their life cycle in order to understand why they're such a pain to try to control. And this is basically the same with all thrips. The life cycle is very quick, 13 to 14 days. The eggs are laid in the plant tissue, so they're somewhat protected from predators and pesticides being inserted into the tissue. You get two larval stages, molt each at about 4 to 5 days. The pupal stages, there's a prepupa and a pupal stage. They last 2 to 3 days. What happens is a certain portion of the population may pupate on the plant, but in general, we're starting to see that most of them appear to be pupating in the soil. So again, they remove themselves from where they might be easily treated with insecticide or with some of our normal predators won't chase them down into the soil. So we have to look at a way to manage them in the soil as well. An important thing that we found is that they don't seem to respond at all to being treated with Talstar. In fact, we can maintain and get a clean colony by treating the plants with Talstar, and it knocks out or prevents some of the other things that we especially the biological controls from eating them. So I'll talk about that again in a minute. 15 eggs per female. Mated female lives 9 days, and adult males live 6 days. Now, some of the growers that have had major problems with managing these thrips feel that if you have an active population, you need to go in on a 4-day schedule with insecticides, especially some of the ones that are contact insecticides. So you need to telescope in your treatments to the point that you're putting them on a chemical on fairly quickly after the last application. And that's one of the only ways that these growers have been able to break the life cycle. As I mentioned, cold tolerance isn't known. We have a scientist, Nicole Benda, working at the Department of Plant Industry in Gainesville for the state, and she's trying to evaluate cold tolerance to figure out how far north this particular pest might be able to establish. We in the greenhouse and in other places, we've tried to talk growers into using indicator plants for the possible presence of this particular pest. Some of the better host plants, it takes a couple weeks at least for thrips to land on it, and actually you start to see some sort of damage. So it can be a lag effect, and we want to get on top of it as quick as possible. So we're using 3 different plants that are relatively easy to grow and that show damage fairly quickly. The photos I'm going to show you in a second here, it only takes 4 days from being exposed to a thrips, parvispinus, for you to be able to see any kind of damage. And these photos were taken at 7 days. This is a bean leaf. Some of the work that's been done on efficacy has been done on bush beans and by Dr. Revinthi down at Homestead. This is Asclepius, and basically you see a lot of damage on the top of the leaf. In this case, we are using Asclepius for some of our biocontrol programs because you can just watch from a distance to see if there's any thrips and if the predators are attacking them. This is a pepper leaf. It shows probably it's one of the better hosts for evaluating the presence of this particular thrips, with the possible exception if you have chili thrips. The chili thrips seem to outcompete them or chase them off. And so we don't find that many of the parvispinus on a chili thrips infested pepper. That may just be coincidence. We don't really know. I haven't had time to investigate that. But so these 3 plants are cheap, they're easy to germinate. You can grow them up to a size that you can put in a greenhouse and leave them for 4 to 5 days. You don't want to leave them there more than a week or 2, or they will start reproducing on you, but they will be good indicator plants if, say, you think you've been exposed to this pest, or if you've had it and you've tried to clean up your greenhouse and you want to see whether or not they're present in your greenhouse again before you start moving some sensitive plants in there. Okay, damage. This is gardenia. They basically look like in a landscape that somebody's taken a blowtorch to them, feeding in all the terminal growth. This particular homeowner said they lost all production of flowers a couple years over the about a 2-year period. They don't get any flowers. The plants are not very thrifty, and they're starting to see damage on other plants near it. This is gardenia again in the environment and basically killing the terminal growth. This is pepper. The adults seem to like the bottom of the leaf. You can't really see the immature stages here. Apologize for the quality of the photo, but again, they feed on both the upper and lower surfaces of the plant. In this case, on pepper, they seem to prefer the bottom. This is a typical plant attacked by parvispinus. Anthurium, you see this yellowing, but generally most of the feeding is on the underside of the leaf. You can get scarring, just typical thrips damage. Hoya, they will kill the terminal growth. This can be caused by broad mites and a few other things, so you have to use a 10 to 20x hand lens, and we recommend to growers to buy a Bosch and Lomb or something, a high-end hand lens. It's worth the money investing in a good hand lens, especially when you have such small critters as a broad mite and the parvispinus. Schefflera, we've seen these in Scheffleras and cut foliage and some other areas in grocery stores. We did find an anthurium that came to us from Canada that was infested in a local grocery store. So anytime you see some unusual discoloration, you see some distortion, you see the new growth dying or just not looking correct, that should give you a warning that maybe you should start looking much closer. And that's what's happened to most of the industry, and why this became such a big threat is that most people weren't looking for something. They thought they had something else wrong other than a new thrips. They're very cryptic and, again, very small. The pepper grower was thinking maybe he had broad mites and he was using something for broad mites, but it wasn't really effective on the thrips, and therefore lost his whole crop. Again, more Schefflera. This is Coca loba. It has been associated damage with the thrips with Cladosporium, Oxysporium, and some other things, and it's been reported to be a vector of tobacco streak virus. But the jury's still out on just how important it is as a vector of diseases. I don't know anybody at this point in time that's really investigating that. We're too busy trying to figure out how to kill them. Chemical management. Okay, we got a couple superstars, one in Canada and one in Florida, that are really doing a lot of good work on management. I think in Canada, this is not considered an invasive species or a quarantinable pest, so they are able to work a little bit more on it than the faculty in the University of Florida. Alexander Ravinthi has done it in almost a closed closet. Okay, it's a quarantine facility where she's not allowed to move any infested plant material out, and she's doing a lot of lab work, bioassays on chemical control, and giving us some very good guidance on what things might have activity in more of a real world setting. She's been using beans and petri dishes, but she's done an excellent job. And she has a paper out, very recent paper out, on her results of her lab bioassays. Now, this is the resource page of my website on parvispinus, and there's two links at the very bottom of the resource page for Sarah and Alexandra's website and some of the work that they're doing. Sarah covers from sticky cards all the way up to chemical controls and biological controls, and Alexandra's doing about the same types of work. We have a grant that we're working on together on looking at biological controls. Again, here's the parvispinus website that you can, if you use your phone, it'll take you to my website, but doing just a Google search of parvispinus will get you there. Okay, some of the work by Alexandra Ravinthi. She's at the Homestead Lab, which is south of Miami. Like I said, she's tested a lot of different materials, and this is some of the things that are showing toxicity. There are a couple others that are generated from some work by just growers, and finding that, some of the OPs actually do have some activity. But it's what you would expect. The thrips materials that work on Western flower thrips, that work on chili thrips, conserve, expire, avid, pylon, all seem to have efficacy at various levels on this particular thrips. Now, they're not at the levels that others have seen on chili thrips and Western flower thrips. It may be just the artifact of the methods that are being used. But again, the management programs for other thrip species seems to work and should work for this particular pest. Now, this is some of the data, and I'll show you. This is too hard to go to in this talk, so a lot of what I'm going to show you is just boiled down clips of some of the things that have been found. Among the conventional insecticides, expire, pylon, and conserve have caused the highest mortality. Pylon is not legal doors. Expire and conserve are basically the same mode of action. So you're not really rotating if you spray conserve and then go to expire. But they do have good activity on this particular pest. There are other materials that have been shown to have moderate toxicity, and they significantly infect the larvae and some of the adults. Now, one of the issues with some of these materials, like I think it's mainspring and pedestal, they're kind of an anti-feedant. And these experiments are run for just a couple days. So if they don't feed, that doesn't but they don't die. Okay, so it takes more than 2 days to die from being treated with an anti-feeding material like mainspring or pedestal. So we may be missing some mortality that would show up later if it were an experiment on a plant outside, for example. But she's done a lot of really good work because she looked at how these compounds impacted the feeding of these thrips, and found that the ones that are anti-feeding, so the plants look good, but the thrips may still be running around. Okay, they may be alive, they may not be feeding anymore, and they're doomed, but we don't know that yet. And she's just started to move out into a greenhouse to be able to run some actual field trials in a more typical type of situation. Overture is proven to be an effective treatment against them. This is, like I said, anti-feeding probably. IR4 put together a sheet on and trying to summarize all the data, looking at giving them ratings of which thrips material has activity on a wide range of thrips, and then rating just their effectiveness. And this is let me say, the PowerPoint that's in there, or that those data were presented, is on my website. So it's a Thrive PowerPoint through Horticulture's HRI Research Institute. They conducted a Thrive workshop webinar, and you can see the PowerPoint presented by Dr. Christy Palmer from the IR4 group. Sarah, she's up in Canada, and she's doing a lot of basic work and looking at IPM as well as just chemical controls. And this is Diplodenia or Mandevilla plants, two different colors, a red one and a white one. White is less susceptible than the red one is. I mean, the red, less attractive. They looked at a sprinch with success, which is basically conserve, and then treating with what we call in the U.S. ARIA as a mid-tank mix, I assume, at 3 months. They got 3 months of activity, so they knocked it down very well. The thrips started to build up on the red, but the white stayed down at a level that they could sell the plants. This is what they consider their threshold for damage, is 10 thrips per plant. Now that's okay up there, and it may be okay down here, but our plants cannot have any thrips on them prior to shipping. So they would have to knock them down with something like Pylon using these techniques and have 0 thrips before they could be shipped out. Now, the different standards for the Canadian growers because of their regulatory issue. Okay, rotation. We try to push that, having at least 3 different compounds to rotate with different modes of action. We're starting to find that we might have enough of a palette of materials that we can mix into a rotation program. Like I said, there's no field data currently available for trials in Florida on Thrips parvispinus, only lab. My lab and Alexander's lab are beginning to do some studies, and we should expect some data soon. J.C. Chong, alumni from Georgia, has now started working for CPRO. He put together a rotation guide that you can get on their website, mixing some of the materials that we know to have activity, conserve, pylon, expire, with some of their materials, Protea and Contos, and basically came up with some rotation programs based on what you're trying to kill, whether it's just thrips adults or thrips immatures, or in a more realistic situation, you're going to have overlapping generations. So you're probably going to want to treat for both thrips and adults. And it has it for greenhouse and nursery. Now you'll notice there is nothing there for the landscape. Landscape has a much more limited palette of materials that they can put together to try managing thrips parvispinus outdoors. Biological control, what's worked for chili thrips, what's worked for western flower thrips, hasn't really worked that well for this particular pest. We feel that this pest is also, as well as chili thrips, could be an induced pest, which means that when you spray some pesticides, you end up with more of a problem. Father of biological control, Carl Huffaker, basically stated that when we kill off the natural enemies of a pest, we inherit their work. And this is what's happening, I think, in the landscape, especially in West Palm Beach, where they have the ability to spray and money to spray on a regular basis. You can go down their streets some weeks and you have to, the traffic is unbelievable. And it's mainly pesticide companies spraying for whiteflies, thrips, scales, a whole array of invasive species that live on the island, West Palm Beach, where Marilaga is. This is a study we ran with ornamental peppers. We had a water control. There's significant damage on the plants, but it's not the worst looking treatment, believe it or not. Mainspring worked fairly well. The new growth is coming out nice. Pylon, again, we had fairly good control and the plants looked fairly well. Some of the damage on and why we terminated this experiment early, some of these plants had broad mites on them, which impacted the population of thrips as well. Conserve looked fairly good. Sericea wasn't quite as good because it didn't have any activity on the broad mites, but the thrips populations were down. Talstar was the worst treatment of the whole bunch. Everything, the plants just died. And the peppers, you could see, were crawling with thrips. There are thrips everywhere. And basically what we found is that we had both a predatory mite and a predatory thrips in that greenhouse that were feeding on the chili thrips and the parvispinus, but the Talstar basically killed them off and the thrips went nuts. We can't in Florida really look at biological control right now because it's still considered an actionable pest and we can't really get to the level that would be acceptable. Future, I think we possibly will be looking at a biological program. There's some issues. They've been looking at this in the European greenhouses. They basically have a problem with this thrips in that the predators, especially the predatory mites, are chased away by the thrips. The thrips will flick their rear ends at an attacking predator, or they'll even flick fecal material at them, which causes the predators to run away and leave them alone. So we've done some work looking at Amblysia swirsky and Lamonicus. We know that this particular predator, the swirsky, will feed on chili thrips, but the work shown from Europe is not real promising. The promising looking biological controls are aureus, where they feel that aureus may have a good shot and may be the most efficient predator for this particular pest. We're looking at Franklinothrips. We have a system that we use, a banker plant system, where we use or pollen and the little tiny sea monkey or whatever you want to call them, cysts that you can buy from BioBee, and we put like a salt shaker and put them onto the banker plant, and this predator will survive and reproduce and maintain itself in the greenhouse, as did aureus. So we had them in our greenhouse all winter long. This is the adult stage. This is feeding on a chili thrips, but they do feed on the parvispinus, and it's too big to be deterred by the fecal droplets or them moving their abdomen to smack the predator. This is the Franklinothrips immature stage, and it's feeding on an echinothrips, so they can eat much more aggressive thrips and larger thrips. None of the other predators work on an echinothrips, but this one does. We tried early on treating plants with lacewing larvae and lacewing eggs. We found that lacewing eggs had an accentuated and probably too high a rate to be commercially viable, but it did work as a management for this particular pest. We're looking at, and so, as well as any of the other entomologists I've mentioned, are looking at soil inhabiting predatory mites, as well as the Dolotia, that's a predatory beetle that lives in the soil. So we're looking for something that will attack the stages that are in the soil. We use a lot of bifenthrin in the soil to manage fire ants, but again, that doesn't deter basically the thrips, because we know when we treat plants with Taustar, the thrips go crazy. We're not sure what it'll do to the predators. Some people are looking at nematodes and insect pathogens. And I know that Alexandra has found that, I think, what is it, Bioseries and a couple other products are looking promising for controlling the thrips. So the summary is the sky isn't falling. We do have effective treatments. Once we found out what was causing the problems, people have shifted their programs and started making the appropriate choices on the materials to use. And I think some are making appropriate choices of what not to choose, which in my estimation is feeling is that Calstar is probably the wrong thing, or synthetic pyrethroids are the wrong thing to be applying. Vegetable growers have more chemical options than the nursery and greenhouse growers. Landscapes and PCOs have a more difficult time due to limited options. But again, over the first year, there's significant losses all over the place, especially in Florida. But they weren't nearly as bad this last year. Scouting is key because you need to detect this early. You don't want to let them get a really good foothold in your greenhouses, and especially if you are dealing with a quarantinable pest. You need to be careful about plant material that you bring into your greenhouses. You don't want to take material to a retail outlet and then go to that retail outlet. If it isn't sold, then pick it back up and bring it back to your nursery. You want to dispose of it properly. And that's it. I can entertain questions.
Thank you, Lance. We have plenty of time for questions.
Okay, I'm going to stop sharing. So I can, there, okay. Sorry I went through so fast, but
It's a lot of information. It is a lot. And one of the things that always fascinates me, and I guess not in a good way, is think about all the invasive species that seem to be proliferating, whether it's a plant or, an insect or anything more advanced, vertebrate. And that's just, we just seem to always be talking and even more and more about managing those pests. And yeah,
it's crazy. I've got at least 4 invasive species out of my greenhouse that we're working on right now.
and it takes a lot to figure out how best to manage them in which situation,
Yep.
greenhouse, nursery, landscape, which geographic region. And the other thing is that they keep expanding. It's like they're moving typically from the point of entry and wherever they established and going northward a lot of times, or southward, depending on the kind of pest. And yeah,
That's why I was getting confused. I keep chili thrips. I had an audience here. And they basically figured out what they're going to do, but I've had to give talks farther and further north on chili thrips, and This week I'm talking on parvispinus. Next week I'll be talking on chili thrips.
Yeah, and I'm pretty happy when I can tell it's a thrips. Going about species, that's a different story. And then one of the things I'm really fascinated with is biological control. Us, we keep telling people to keep on, especially homeowners and in ornamental landscapes, and don't reach to any chemicals. Just let nature take its course. But a lot of it is just knowing what the natural enemies are like in different stages of life of an insect and or a spider, and figuring out how you could protect those as much as possible. Yeah. Grace has a question, which is not related necessarily to what you spoke, but I actually had that question too. What kind of wasp is on your background?
I don't know. I just took it out of my front yard. I'm not sure what it is. A Vespa, I think. But if my job depended on me identifying so many insects here in Florida, I'd probably be working at McDonald's.
You're too modest. But then yes, it's remarkable what a great variety of insects and arthropods an entomologist must know. I'm always surprised when I ask them, what is this? And they like, I don't know. I'm like, what do you mean you don't know? So the Asclepius that you use, you said that the thrips is attacking it as well, because I always consider that the biggest thing is aphids, oleander aphids, kind of like these go hand in hand.
We use a lot of Asclepius here. We use it for banker plants for aphid predators. And so that's how we found that the parvispinus was an issue. So now we use a lot of banker plants. Now we're having to find natural enemies to release on the banker plants to keep them clean. Okay, so the oleander aphid, when we can't get it near the mandevilla or the diplodenia, or it'll take out all the new growth. So it's got a lot of balls in the air, but we use them because they do show damage really quick before the aphids can really get going on them.
Yeah, I mean, I have I don't grow the tropical one that you have there, but I grow the other species, at least
Mm-hmm.
6 species in the landscape, and it is remarkable to me to see how early the aphids are. I mean, I could actually find aphids on something that is honestly like half an inch almost under the mulch that has
Yeah.
some green tissue, and there are the aphids, kind of hunkering down, just waiting for that new growth to start, and they just coat it. But it's a great opportunity to observe natural enemies.
No other questions, huh? Yes.
I guess it's just a shy crowd today, or they were overwhelmed. So I encourage everyone to check out the links that Lance shared with us. There was a lot there, and I'm sure that the information will give you plenty to think about. And if you have any questions, you can reach out to him directly. We do have one question. Is malathion effective on thrips?
On some species, I think it is. Nobody's really looking at the OPs for this particular pest. Not that many OPs or carbamates are used in the ornamental industry in Florida, as far as I'm concerned. We did have some growers that have used some of the older compounds. Some were not illegal to be used, and they did have some impact on the thrips. But I think we have better options, safer options.
Well, if there are no more questions, thank you, Lance. Thank you, Anthony. That was a great information. And thank you all. We'll see you in a couple of months.
Thank you.
Thanks for having me.
Bye now.
Bye.